Dames en heren, gaat u zitten. Dames en heren, geachte collega's, ik heet u van harte welkom op de openbare doctoraatsverdediging van Armen Tchobanian en ik verklaar de zitting voor geopend. De heer Armen Tchobanian, Master of Science in Molecular Biology from the Systems Biology at the University of Montreal, Quebec, heeft aan de faculteit een proefschrift voorgelegd, handelend over Polysaccharide Esters for the Topochemical Design of Biointerfaces. In het Engels, in het Nederlands, Polysaccharide Esters voor het topochemische ontwerp van biointerfaces. En dit tot het verkrijgen van de graad van dokter in de ingenieurswetenschappen. Hij zal dit proefschrift nu verdedigen. Ik vraag aan de kandidaat eerst een uiteenzetting van ongeveer 45 minuten te geven over zijn werk. Daarna zal er gelegenheid zijn tot het maken van opmerkingen en het stellen van vragen. You have 45 minutes to present your work, after which there will be a question and comment session. Meneer Armen Tchobanian, u heeft het woord. You have the floor.
Thank you. Can everyone hear me? Good. And I hope everyone who's joining us online can hear me too. I'm obviously Armen, as you should all know now since you've been invited to my defense. And I'd like to first thank the jury for being here today. And I'd like to welcome the audience that is with us here in person and those that are joining from the safety of wherever you may be. So over the next little while, I'm going to describe the research that I've been performing throughout my PhD on the topic of polysaccharide esters for the topochemical design of biointerfaces. And hopefully by the end of this, you'll be able to identify and understand what a few of these words in the title means. But before I get into my research, I need to set the stage a bit within the context of two larger fields. And these are regenerative medicine and tissue engineering. These are both relatively new fields and multidisciplinary fields that began in the late 80s, early 90s, or began to emerge in those years. And they aim to replace, repair, or regenerate, obviously, cells, tissues, or organs in order to restore or improve their function. Now, the idea of regenerating tissue isn't a new one, and I think perhaps the most common example of this is the punishment of the Titan Prometheus. And so this Titan was punished for giving mankind fire, and his punishment was to be chained to a rock and have his liver eaten out by an eagle every day, and every day the liver would regenerate, his wounds would heal, and he would suffer the same fate the next day and the next day for eternity, or at least until I believe Hercules helped the guy out. There is another reliable source of information other than mythology for this regenerative tissue, and that is sci-fi, where you have devices that can heal wounds instantaneously or regrow limbs or organs. And these are nice ideas, but they don't exactly cover what these two fields deal with. And I'm going to focus specifically on tissue engineering. And I'll begin with the original definition of the field that was given by this man here, Robert Narum, at a workshop held in 1988 to identify appropriate areas of research in this emerging area of technology. And his definition is as follows. It is the application of the principles and methods of engineering and the life sciences towards the fundamental understanding of structure-function relationships in normal and pathological mammalian tissues and the development of biological substitutes to restore, maintain, or improve function. Now, you may not understand what everything in this definition means, but hopefully you can identify that there are two different colors of text here. And the reason I put this here is because 5 years later, two other prominent figures in the field wrote, I believe, the most cited review paper in tissue engineering, and to this day, I believe it has over 7,000 citations. And they quoted his definition, but it became this. And these gentlemen are Robert Langer and Joseph Bacanti. And this is not to say that people are no longer interested in researching the fundamental basis of structure-function relationships in tissue, but it does kind of mirror the focus of the field in general. Most of the research is focused on developing functional biological substitutes, and this is not necessarily wrong in any way. But what is tissue engineering in practice? Well, it focuses on this thing down here, and we call this a scaffold. It is a 3D material that is constructed of various biomaterials, bioactive components, and often cells. And it is designed in a way that it can support the regrowth and regeneration of tissue. And that is the idea, at least. And what you see here is the life cycle, or the proposed life cycle of a tissue engineering scaffold or implant, wherein you isolate cells from the patient in the area that needs to be replaced, you expand them, and then you design the scaffold to support the growth of this kind of tissue, culture the cells on this material, allow the tissue to begin developing over a course of a week or 2 weeks, and then implant it back into the patient, where the idea is that it would support the regrowth of the given tissue that is designed for, and over time be degraded, leaving nothing left over but the functioning new tissue. And probably the most popularized example of this is shown here. And it is also one of the earliest, I believe, as well. And so what you have on the left here is a scaffold, and it is in the shape of a child's ear. It is made of a polymeric material, but that's not quite so important at this point. And it has been seeded with these cells known as chondrocytes, which are cells in your cartilage, like you have in your ear. And they then implanted this material, seeded with the cells, into the back of a mouse. And what the researchers found is that over the course of several weeks and months, this environment was able to support the regrowth of new cartilage, of new cartilaginous tissue. So you may wonder, well, how exactly does this work? How can you take cells, put them on this scaffold, stick it in a mouse, and expect it to regrow the shape or the type of tissue that you intend to regrow? Well, it's rather complicated and not entirely clear to us at this point, but I will try my best to explain it to you. And I debated how to begin this. And I thought initially I would start like this, with the standard bathroom symbols that we have. But I thought perhaps some of you might complain about that. You might say, well, my head doesn't float above my torso. I have more than one leg. So I thought I would borrow an example from physics. And I will say, this is you. And to be precise, I'm assuming that you are this sphere, of course, in the current environment. And if we were to look inside of you and look at the various kinds of tissues you have, for example, your muscle or an organ or bone or skin, you would find that depending on what kind of tissue you are examining or even the location in the tissue, you will find different kinds of cells. And these cells have their own critical functions to carry out that allow me to stand up here and tell you that you are a sphere. But that's not all that's in your tissue. There is a lot of material surrounding these cells, and this material can vary in its composition, again, depending on the tissue, and this is precisely what these tissue engineering scaffolds are meant to replicate in their structure. And this material is appropriately called the extracellular matrix. So it is all of the material external to the cells in your tissue. And we are interested in this environment because it provides many of the biological cues, whether they be biochemical or biomechanical, to direct cell behavior and fate, and can support the regrowth and regeneration of tissue by means of that. But it is also continuously remodeled by the cells themselves in order to regulate this behavior in sort of a feedback loop. And what you see here, although you don't necessarily need to read it, are examples of some of the components that exist and where they may exist in relation to different cells, such as those up here or those here. And although this is a slightly more sophisticated image than my confetti from the previous slide, it is still a vast oversimplification. And I won't ask you to read this later because I don't even think I can read it right now. But what you have here is a list where every row is a different identified component in the human extracellular matrix, and they number to roughly 450, and these are the core components. So it's a very diverse environment. And in every column at the top, you have either a different tissue or a different state of a tissue. And if a box is colored in, the component exists in that tissue. So what you can see here is not only is the environment very compositionally diverse, but depending on what kind of tissue you look at, and depending on even the age of a tissue, any history of injury or disease in the tissue, this composition can vary to quite a large extent. But if you were to group the most common components in this environment, you would find that many of them are proteins, polysaccharides or carbohydrates, and these two columns on the very left here, which are called glycoproteins. And these are effectively proteins decorated with little carbohydrate fragments. And these complicate the issue quite a bit more. They make up something known as the glycome. It is the complete collection of carbohydrates in an organism, in this case, us. And I have an example of one over here. It's this little symbolic diagram decorating an example protein over here. And we know that these carbohydrate fragments are critically involved in many biological functions, but unlike your proteome, which is the entire set of proteins in your body, which is encoded in your genome, these structures are not. They are created by a sequence of enzymatic events. And because of this, it is very difficult to predict what their structures may be, predict where their locations are. It provides quite a bit of heterogeneity into both of these aspects, and it makes it rather difficult to decipher what their means of interaction is. All of this is just to say that it is a remarkably complex environment, and the replication of this composition and all of its functions is not a simple task. So if it's unrealistic to replicate such a complex compositional environment, then we can look at trying to replicate some of its major functions. And there's a lot of different ways the extracellular matrix impacts cell behavior, and a lot of different properties it has that can do this. And so what I have here are a few properties of the extracellular matrix that can directly have a profound influence on cell behavior. And I'm going to start with these two at the top here. And I'll consider these more biologically oriented forms of influence. So you can have recognition sites or different biological components in the extracellular matrix that cells recognize and will adhere to. And you can include these same components, for example, in a synthetic scaffold. You also have different components in the extracellular matrix, such as these growth factors here, that can stimulate cells to develop in a specific way or to behave in a specific way as well. And then you have a lot of other properties that are not so, say, biologically conventional. And these are different physical and chemical properties of this environment. So for example, the different chemical groups covering the material can influence, in a way, how cells behave when interacting with it. But also different structural features such as the porosity or density of a material and mechanical properties such as the stiffness or softness of the material can all impart an influence on cell behavior. But the problem is, even though we are aware that many of these different properties, even some that aren't included on this figure, have a profound impact on cell behavior, the precise mechanism through which they have this influence is not entirely clear to us. And what makes it even more difficult is that at this moment, and I will say to the best of my knowledge, we do not have a reliable way to construct three-dimensional materials with enough control over all of these properties at the scale at which cells respond to them. So it's a very difficult task to properly model something with all of these different properties, or to control all of these properties. But they are rather interesting, in that you could design a synthetic material to have many of the beneficial properties through these means. So one thing we can do is to slightly simplify the situation and look at a surface through which many of these same properties can have an impact on how cells interact. And so you see again for example, softness or stiffness of a material, various chemical influences from the surface of the material, and some topographical influences. And there has been quite a bit of study to see how many of these parameters, for example, can influence how a cell interacts with the surface. But I would say, to understand the mechanism behind it, the jury is still out. But that doesn't mean they're unimportant or uninteresting. And here I'm going to focus on two in specific. So the surface topography shown here, and the hydrophilicity or hydrophobicity shown here. And I will group some of the properties that would be collectively under these two categories in surface energy. And this is effectively the measure of excess energy present at a surface of material in comparison to the bulk. And even if that doesn't make sense, it is a property that can influence how different materials interact with one another. For example, how this water droplet interacts with the surface of the leaf. It is a rather interesting property of materials in that it can be influenced by both a physical and chemical component. So the surface chemistry or the chemical composition of a material can influence the surface energy, but so can the roughness of a surface. So if we take the same example of a water droplet on a surface, based on its interaction with the surface, you could broadly categorize it in many ways, but here I have shown two of the most common, and this is a hydrophilic material. The water likes to wet the surface, and a hydrophobic material down here where the water beads in a way on the surface. And we can characterize and measure this wettability through something called the contact angle, which is shown here with how the droplet interacts with the surface. So if we were to take these two, say, chemically uniform and planar surfaces, and then introduce roughness into them and examine them again, you would find that this material over here becomes apparently more hydrophilic, and this material over here becomes apparently more hydrophobic. And the interaction between these two properties is rather interesting, but especially in the case that they're not often examined together in the context of cell or protein surface interactions. The last property I'll introduce is somewhat related to roughness, but we'll call it surface topography. And I will differentiate them by saying this is a larger scale of surface information, whereas you could have roughness overlying a topography. And this is probably one of the earliest influences that was observed in material interaction with cells, and it was observed over 100 years ago, I believe, at this point. And the surface topography can impart an influence known as contact guidance. And in essence, what this is a mechanical interaction between the cell and the surface that is able to influence a cell's orientation and even its shape or morphology. And this is quite interesting because simply through the alteration of a cell's shape, you can influence how it behaves and even how it develops. So together, these and all the other properties that were shown before are quite interesting to investigate in the context of cell surface behavior. But you need a material that can be controlled precisely enough in order to control the properties of the surface and the topographical patterning of the surface as well. And there is a group of materials that is quite interesting in this context, and these are polysaccharides. And they are interesting for quite a variety of reasons. Polysaccharides are naturally derived, abundant, and renewable materials, which is one benefit. And you can find them in many areas in nature. For example, in different plant matter, you can find polysaccharide like cellulose. In different crops, you can find starches. You even have microbially produced polysaccharides such as dextrin and pullulan. In the exoskeleton of crabs and different crustaceans, and even in some fungi, you can find polysaccharides such as chitin or chitosan. And in some aquatic plant life, such as algae, you can find polysaccharides like alginate and carrageenan. But these naturally derived polysaccharides are also interesting because they are, in a sense, structurally analogous to many of the natural polysaccharides we have in our extracellular matrix. And this imparts them with some beneficial properties. And in general, most of these naturally derived polysaccharides are biocompatible, so they don't influence in a negative way our immune system or impart any kind of toxicity. But they're also, in general, biodegradable in our body, and they can be broken down as you would want in a tissue engineering scaffold material. But the reason they are precisely quite interesting in this context is the diversity of structures that can be produced with them, and thus the diversity of functions. And here, I have an example of this. Don't be scared of the slide. So polysaccharides are polymers of monosaccharide units. And I have an example of a monosaccharide unit in the middle here called D-glucose. And these typically exist in nature in a cyclic or ring structure, as you can see these four at the edges. So through this one monosaccharide, you can have different cyclic structures. But each of these monosaccharide units can also interact with itself and with other monosaccharides in a variety of ways through glycosidic bonds, and I have just shown 3 examples down here. So even with one unit, you could create quite a large combinatorial variety of structures and thus functional materials. These polysaccharides are also quite amenable to different chemical modifications, which is quite interesting as well. So I was interested, how are polysaccharides being used in the context of tissue engineering? And I looked at that. And I read quite a few papers, more than I would have liked to. And then I did a form of meta-analysis, where I extracted text-based information from roughly 11,000 articles on the use of polysaccharides in tissue engineering. And what you see in front of you is an example of some of that data. This is what would be called a keyword co-occurrence network. So these are the 100 most common keywords extracted from all of these articles. And depending on the size of the bubble on this diagram, the more frequently the term occurs. And depending on the thickness of the link between two, the more frequently they co-occur. And I saw quite a few interesting things in analyzing some of this data. The first is that there were 4 predominantly used polysaccharides in the field that make up over 70% of the research. And these are chitosan, shown here, cellulose, alginate, and hyaluronic acid. But what was more interesting is the way these polysaccharides were being used, and this was a rather simplistic manner, I would say. In general, most of these polysaccharides were used effectively off the shelf from some commercial supplier, and then synthesized into a 3D structure, such as a hydrogel, shown here, and then tested to see how it behaved or how cells would behave on this material and if it would be an effective material in this context. And this is not an ineffective way to approach tissue engineering. And in fact, in a lot of the cases, this is quite effective in its outcomes. However, it sort of sidesteps the understanding of how the structure of these molecules and how the orientation in the developed scaffolding is able to create such an efficient structure or an effective structure. And so that's where my research came in. And the main aim of my research was to design a biointerface with polysaccharides. So I wanted to reliably produce polysaccharide-based model platforms with control over multiple surface chemistries that are relevant to cell surface interaction. And once this platform was designed, I wanted to see first if it was even suitable to be testing with cells, and if they would attach and grow on the material, but then to see if this kind of property-based design could have potential to be used as a platform for investigating cell surface interactions. So obviously to do this, I need a polysaccharide. And for that, I chose dextrin. An example of its structure is shown here on the very right, this linear polysaccharide with various branching points along it. It is a microbially produced polysaccharide, and it has quite an interesting property to it in that unlike most naturally derived polysaccharides that are commercially available in a very heterogeneous distribution of sizes or molecular weights, dextrin is available commercially in very narrow molecular weight distributions or very narrow size ranges. And this makes it quite interesting because then you can have a very uniform starting material for producing a very precise control over a structural property of a surface. It is also soluble in a wide variety of solvents, and this allows it to be chemically modified through a variety of routes, which is quite interesting as some polysaccharides are very difficult to dissolve, for example. And lastly, but not least, of course, dextrin has already been widely applied in the medical field and has quite a well-studied physiological activity in our body. So this is a polysaccharide, but then we need to introduce control over some of these properties. And to do this, we come to the polysaccharide ester part of that title, and we esterify dextrin with a fatty acid known as palmitic acid. And what you see here is the synthesis route that I had used to create this polymer, and it is shown down here with an ester bond between the palmitic acid and the dextrin. And this was a rather robust synthesis route in the sense that it could be performed in a single vessel with 2 steps. It had very few byproducts which were non-toxic, and the resultant synthesized dextrin palmitate here was very easy to purify out of the reaction mixture. So why would we want to put palmitic acid on this material? Well, there's 2 reasons. And the first is to alter that hydrophilicity or hydrophobic balance of the material itself and thus a surface designed with it. And dextrin on its own natively is a rather hydrophilic material, so water will wet it very well. But also the introduction of palmitic acid onto these molecules produces a thermoplastic material. And this is effectively a material that you can melt and set into a structure. And this allows it to be suitable for a variety of patterning processes that we will see later. So now we introduced one form of control, the surface chemistry-based wettability. Then we need a surface. And to do this, I used a method known as spin coating. And this is a method derived out of the field of microfabrication, where essentially you produce microchips that you find in your computers. And it's a very robust film forming technique. And what it involves is the dispensing of a solution of your polymer onto a surface that will support this film. This is then held on a rotating mount. And depending on the rotational speed of that mount and the properties of your solution and the environment, you can very precisely control the thickness of the film you produce and the uniformity of the film. So it's a rather robust film forming procedure. But now, of course, you need to introduce some topographical information into the surface. And to do this, I looked at the same field and used a method that is not commonly used with polysaccharides or even in the field of tissue engineering, and it is known as thermal nanoimprint lithography. And I'll try and walk you through the steps of this procedure. And it begins as most chips are patterned, where you have a silicon wafer that is patterned with a given topography and then cleaned and purified at the end. This is then transferred into a rubbery stamp-like material to obtain the negative imprint of the topography. And finally, this stamp is placed on top of your freshly spin-coated thermoplastic polymer now. Weight and pressure is applied, and the heat is raised to allow the material to flow into the stamp as the stamp sinks down into the material. Then the temperature is lowered to allow it to set. The pressure is removed, the stamp is removed, and you should have effectively transferred the pattern from your silicon wafer, which we call a master, to your polymer coated on, in this case, a glass surface. So these are the three major steps to designing this platform: the synthesis of the material, the forming of a film, and then the patterning of the film. But of course, to pattern something, I need some kind of pattern. And so I looked to see what was being done in the literature. And this is specifically research done with topographical influences on endothelial cells, which would be the cells I would examine later on. These are the cells found lining blood vessels. And so what we have here is a chart of the feature size of a topography versus its spacing. And what I mean by that, if you look at the diagrams over here, the feature size is simply the width of a feature, and the spacing is simply the width in between two features, or the space in between two features. And so in looking at this, I found a few interesting things. The first is that the vast majority of research is done with topographical patterns that have equal feature width and feature size and equal spacing, which is not necessarily a bad thing, but also that there's a very large feature space, not feature spacing, but combination of sizes and spacing that haven't been examined that could have an important influence on cell surface behavior. But I think most importantly is that there are certain dimensions of a topography that cells will not readily respond to. And to give an absurd example, say, is if you have a topographical feature that is a few orders of magnitude larger than a cell, the cell interacts with it as if it's a surface, not as if it's a topographical form of information, unless, of course, it reaches the edge of that feature. And so based on all of this information from the literature, I selected the three most simplistic and primitive forms of topographical information, and these are gratings here, these linear structures, pillars, and pits. And then I had organized these into different grids up here, alongside an unpatterned control. And these grids are arranged as follows on the right. They're arranged with a variety of feature sizes and a variety of feature spacings that are intended to cover from the multicellular range to the single cell range to the subcellular range. And here you have 81 different combinations of all of these properties. So now with the pattern, I could continue with what exactly I did. And that starts with the pattern, or the synthesis of the material. And so I synthesized a variety of dextrin palmitates, shown up here, and here's an example of what a structure of that might look like. And I did this with a few different molecular weights of dextrin, varying the molar ratio of the reagents in the reaction in order to yield products that have varying degrees of substitution. And what this degree of substitution is effectively the number of these three regions on the dextrin backbone that are replaced by a palmitic acid group. So the maximum degree of substitution you could have is 3. And the reason we would want to have various degrees of substitution is to be able to theoretically be able to control the hydrophobicity or hydrophilicity of the material in the end. I then did some chemical analysis of the synthesized compounds to ensure that it had the intended structure and composition, and that it was pure enough to be used in a biological setting. So these are results from something known as an FTIR analysis and an NMR analysis. I characterized the thermal properties of the material, of course, to ensure that it had the necessary thermal behavior to be used in the intended patterning process. And finally, I spin-coated some films of these different materials and examined how they would alter the wettability or hydrophilicity of this material through their composition. And this is done via contact angle measurements with water, and also calculated the work of adhesion that correlates to this between the water and the surface. So with the compound synthesized, I could continue on with the patterning. And this, of course, begins with the silicon master shown up here. And you have examples of the different structures that appear on this structure, or that appear on this silicon master through scanning electron microscope images. And these colors you see in all of these three figures are simply the diffraction of light with these small structures on the surface. So once I had successfully patterned a silicon master, after verifying the dimensions of the features were what I intended them to be, I could continue on with the stamp made of a material known as polydimethylsiloxane, or PDMS. Again, verify that the negative imprint was transferred efficiently. And then finally, I needed to figure out how to pattern my dextrin palmitate films, as we didn't necessarily have a device to do this. And I used a very simple setup, which involved a heated plate, above which I would have my spin-coated material, above which I would have the stamp, and then a heavy steel weight on top of it all. And that's how I would apply the pressure to the system. And of course, this simplistic setup needed quite a bit of optimization to reliably transfer the patterns from the stamp to the surfaces of the spin-coated materials. And after quite a bit of time, I did manage to get it to work quite reliably. And here you can see the pattern films on top of a glass substrate set inside a cell culture plate, and some examples of the structures there. And to examine the topography of these materials, I use a method known as profilometry up here. And here you can see examples of a few of the structures. You have the pillars, the gratings, and even if you can't identify them, these are pits. And I include this for a particular reason. This is one of the difficulties I came across in doing this patterning procedure. So effectively, because I have such a wide range of sizes of structures on my material, it becomes difficult to flow the material into the larger structures at the same rate as they would in the smaller structures, for example, here. So what the issue here is that the material isn't completely filling the cavity or void in the stamp. And of course, this could be optimized in a way by altering some of the parameters of the procedure, such as increasing the pressure, increasing the time at which the procedure is carried out, and so on. But in seeing this deficiency in the procedure itself, I saw something else rather interesting, and that was a difference in roughness between the areas where the material flows, such as in these pillars, to the spacings between them. So what I mean by that is the tops of these pillars here had a significantly increased roughness compared to the bottom of the pillars, and the same with the gratings, and of course, the same with the pits here. And I thought perhaps this could have a large impact on the properties of this material itself. And so I investigated to see how this roughness would contribute to that. And what we have here on the very left are the contact angle measurements of the unpatterned and patterned films. And you can see that they are both quite hydrophobic materials at around 111 to 112 degrees. And then I want to see how the roughness of the various regions in these patterns could influence this property. So how it would influence the apparent contact angle of the material in a sense. And if you look here, we have the planar regions of the material, and all the lower regions of the patterns have a very similar property as the film. In fact, they're not significantly different in any way. However, these areas where the material would flow, for example, the pillars, the gratings, and the spacing between the pit were significantly different. They had a rather significant impact. And even though this is on the order of 1 to 3 degrees of the contact angle, this could have quite a large impact on cell behavior. But then I need to make sure first that these cells would even appreciate all the work I put into these materials. And so I did just that. And what we have here on the very left is a control, which is a surface coated in a protein known as fibronectin that cells will attach to. And then the right 3 columns here are different dextrin palmitates I have synthesized at various molecular weights. And what I had observed was that the cells were able to attach quite effectively to my materials. They had the morphology that I had expected, and that they were able to grow and proliferate rather readily. And when comparing the situation between the control and the different materials, there was no observable difference in these preliminary assessments. So at this point, I thought, okay, the cells don't hate my material, so I can see how these different surface properties are able to influence, in combination with the topography, cell behavior. And that was quite interesting. So first, what I have here on the top right, in case anybody is confused, is a well in a cell culture plate. At the bottom, you have the glass substrate upon which I have the patterned dextrin palmitate structures. And then the cells are cultured on top of this, and it is all submerged in the appropriate media to allow the cells to survive and grow as they would. And what we see first is that the planar portions of these patterned films impact or elicit a very similar response on the cells as they would on the unpatterned films, which is effectively what we would expect. But then the cells had a rather interesting interaction with the patterned regions of the films. And what I was observing was that the cells seem to attach in the regions of lower roughness in these patterns. So in between the pillars, in between the gratings, in these grooves, and in the pits or between the pits like that. And what was even more interesting was that it seemed that depending on the size of the features and the spacing of the features, there was an influence on how the cells would interact with the surface. So for example, when they are close enough with the pits, the cells seem to bridge across from one to another. If they're large enough to fit one, they can get trapped inside. And here you can have different multicellular arrangements growing around the pillars themselves, for example. But I need to make sure that, well, for example, my eyes aren't deceiving me. And I did some image analysis on all of the different microscopy images I had taken to verify what I was observing. And what we have at the top here is an assessment of the fraction of the cell area that overlaps with these smoother regions in the pattern. So in between the pillars, in between the gratings, and in between the pits, or the pits themselves, rather. And what I had seen is that an overwhelming fraction of the cell area did overlap with these regions of the patterns, which could indicate that there is an overwhelming frequency of cells to want to adhere to these regions, or a tendency to, rather. But what I also observed was that there was a quite profound influence on cell alignment. And so what we have here on the left is with the pillars, the cells are prominently aligned along the 0 and 9 degree axis, so the grid defined by the pillars. With the gratings, you have a very overt alignment along the length of the gratings. And with the pits, it's a bit more unclear, but based on the microscopy observations that I had, the pits allowed the greater amount of different orientations to the cells. So the cells could effectively sit inside a pit at differing angles, but they could also bridge from one to another depending on the size and spacing of the pits. So it's not necessarily that unexpected. finally, I needed to make sure that there was any point in making all of these combinations of topographies, 81 different combinations for every different feature type. And so I looked at how their spacing and their sizes would influence some of the cell behavior. And so what we have here are an assessment of the feature size and spacing of each of the different types of features, the pillars, the gratings, the pits, and their influence on cell alignment angle on the left here, and cell aspect ratio on the right here, which is effectively the degree to which a cell is elongated or rounded. And although I will say that it is far too early to concretely indicate that a certain, for example, feature size and spacing is the optimal selection for influencing a cell to take a certain alignment, this does give quite a few glimpses into what this platform could have a potential for. But what I also observe in these figures is that there is a very clear difference between how the cells interact with each type of feature, specifically, but also how the influence of the feature size and spacing may be on some of these cell surface behaviors. And so this approach to designing material surfaces through a control of their properties alone could be a rather interesting route in assessing some of these cell surface behaviors, if it was to be developed further and optimized further. So, in conclusion, I developed a patterned dextrin ester platform using a simple in-house procedure with a hot plate and a steel weight. I performed thermal nanoimprint lithography on polysaccharides for a biological application for the first time, and I believe also for the first time for dextrin. The developed procedure was able to yield a platform with control, or relatively good control, over the synergistic properties of surface chemistry, surface roughness, and surface topography, although the last two properties were linked together through the patterning process. The designed films were able to support the growth of endothelial cells, as well as the standard fibronectin coatings. And finally, these patterned films were able to present restricted domains of cell attachment and alignment simply based on the combination of these different surface properties themselves. So I would like to acknowledge the people listed here for their various help throughout my research, and I would like to thank you for your attention. Should we hand around a microphone or? Okay, okay.
There are two external members who are not present today, but who participated in the preliminary events. And then Professor Karin Stana-Kleinscheck from the University of Graz University of Technology in Austria, and Professor Jolanda from Hengel from the University of Ghent. Who are present today are Liesbet Geris from the Department of Chemical Engineering and Professor Pedro from Pyyvelde, Department of Chemical Engineering. And they will now ask different questions and make their comments. And for this we need the microphone. And the first one is Professor van Pyyvelde.
Hello, Armen.
Hi.
Thanks for your presentation. Nice overview of your work at the end, it turned out to be a relatively thick book. So
yeah, apparently.
I got it a little bit late, so I did not have a lot of time to prepare questions. But nevertheless, I have 3 of them for you. Okay, now you have accomplished your work with some biological polymer, bio-based polymers. Let's say, okay, it's a bit in between since you use standards, but let's call Dextran something that can come from a biological source, let's say. And at one point in your literature review, very briefly on page 5, you give a paragraph where also synthetic polymers have been used. Now, of course, synthetic polymers have some advantages in the sense that you can control molar mass and the like in a very reproducible manner. So it's basically whether you make it today or next year, it will be the same, which is obviously not the case if you use materials from a biological source. And my question actually is, so you mentioned that some of these polymers have been used in scaffolds, and you refer to sometimes the lack of biological recognition. But if you think about materials like polycaprolactone and things like that, they have been used in the past. So what's your added value with respect to these kind of synthetic approaches? And what's your main selling point, your unique selling point for your materials with respect to the synthetic counterparts, let's say?
So, in general, I would say one of the big benefits of using these naturally derived materials is that renewable aspect of them, but also that we're getting closer in a sense to synthetic bacterial pathways. So you could, in essence, control these polymers that it's producing, these polysaccharides. But they can also be modified very precisely as well, and don't need to be inherently coupled with other bioactive recognition capabilities or bound in a specific way to allow them to be degraded by the surrounding cells. So I see that as one of the major benefits.
Is this something you cannot do with the synthetic alternatives?
You can, but it is still, I would say, rather challenging to avoid some of the problems of their degradation as well. So the byproducts of the degradation can occasionally be impactful on the body. Whereas the degradation products in general of most polysaccharides are relatively inert and can be removed.
All right, okay. My second question is more towards the economic evaluation of your method. So whether in the conclusions or in the future work part, you refer to the 80 cents per square centimeter for the material.
8 cents, I believe.
80, I think.
80?
No? 80,
Okay.
0.08, yes, 80 cents, yes. But of course, that's only for the material. So the thermal nanoimprint lithography, okay, you mentioned in your conclusion here that it's also cheap.
Yeah.
But still, there are probably some upscaling issues that need to be taken into account in order to make this economically viable. And can you dwell a little bit on that?
So I would think if you were to scale it up to an industrial perspective, the biggest challenge would be to, one, find a suitable stand-in, for example, for the silicon wafers. So you could design a rolling plate, for example, with the patterns in it that is designed out of a metal, for example, that could continuously produce these patterns. That's also usually one of the difficulties is the fragility of silicon itself and the costliness. If you do not have the facilities to produce it, the initial cost investment in the pattern silicon wafers can be quite high.
And do you see any issues with the synthesis scalability? Because, I mean, it's pretty hard to tune towards 100% conversion.
Yeah, that's one thing that I wanted to look into further, and I did mention as well in the conclusions. But I'm not sure, I haven't come across large-scale uses of this synthesis pathway, but I think it could be quite viable, at least to an extent.
Okay. And then the final question is related to, of course, one of the most important issues, that's the interaction of the surface with the cell. So the surface properties with respect to the cell behavior. And you mentioned surface energy so surface energy, and to work on surface energy in order to increase this interaction.
Yeah.
Well, surface energy, there are different components associated with that. You have the dispersive parts, you have the polar parts of surface energy.
Yeah.
So I have let's say, a question that consists of two parts. So first of all, what would be your approach? Would you work more on the dispersive parts or more on the polar parts? And then secondly, what kind of modification technique are you envisaging? Are you planning or are you suggesting to work more towards the chemical modification by really chemical synthesis approaches? Are you referring to the use of etching techniques? Are you referring to, forgive me the word, corona techniques that can be used to treat surfaces? Nothing related to it, the corona that is going on here. Are you thinking about certain additives? So what would be your approach to that problem?
Well, in this sense, what we have done here is more of the, I would say, dispersive side of that influence on surface energy. But I think it would be also interesting to see if you were to introduce also polar components, how they could work together for a specific kind of influence or independently.
Did you try that during the, no?
No, I didn't have a chance to try that.
And from the technique point of view?
You can couple in a variety of components in tandem with the synthesis technique, though it could be quite difficult to control the degree to which both of them couple the material unless you protect certain functional groups. But I would have to look further into that to see how that would be applicable.
Okay, thank you. Thank you, Chairman.
Thank you.
Thank you, Armen. Thank you for, or congratulations with a very entertaining and eloquent presentation. The same as in your book, you make it easy to follow.
Thank you.
I'm going to start with a question that I think I've asked you almost every time that we met, and that is going back to the frame of the tissue engineering that you used in your thesis. Now, you've shown a proof of concept here.
Yeah.
And so let's think ahead a little bit and think about some of the challenges that you will face if you want to implement this in a tissue engineering situation. And so some of these challenges relate to the cells that you need, the biological functionality, the cost, the biocompatibility, upscaling, and regulatory, some of which have already been addressed now. So let's start with the cells. Of course, in tissue engineering, you combine materials and cells, but you could also think that what you are doing is maybe suitable in a cell, just any kind of cell-based approach. So not necessarily a tissue engineering approach, but just cell therapy. So do you think you necessarily need to use your matrix to create a tissue engineering construct? Or how could your strategy help to improve cell culture, for instance?
That's a very good point. I wouldn't necessarily use this specifically for developing a tissue engineering construct. The general idea behind it was to sort of rationally design a platform with which you could hopefully better understand some of the interactions between these properties and cell behavior, but this could be applied in a variety of different applications, not just tissue engineering. So I think it's quite a translatable form of platform in terms of the results it may produce.
So if you think specifically about cell culture, which can be either 2D in flasks or it can be 3D on beads, how do you see your results integrated in such a platform, or the things that you investigated?
Perhaps in a setting like that with the beads, I would not see it as simple to include some of these properties in that sense, but where I could see this being applied is, for example, in a setting of a bioreactor where you could have it coating surfaces within the reactor that the cells can circulate around and through for kind of a larger scale setting.
Sorry, I didn't really understand the last. But so in a bioreactor setting where the cells can...
Well, for example, if you had, if you could imagine, a coil inside a bioreactor, and the surface of this coil is similarly patterned.
Okay.
That kind of idea.
And so that would be a way to apply this in 3D.
Or in a larger scale, at least.
Yeah. Okay. And when you then want to go to 3D functionality, increasing biological functionality. So you use the example of vasculogenesis, of angiogenesis. Is there a way to integrate what you've done in a 3D construct? Is there just any kind of way that you can think of to try and stimulate blood vessel ingrowth in a 3D construct using some of your technologies?
So one way would be kind of similar to what I was mentioning before, but you can remove these films from the surfaces and form them into different kinds of structures. For example, if they were to coil up in a more tubular structure. But I think also it could potentially be used in a similar way, but I imagine this would be quite difficult, is to layer different patterned materials together. But then you come into difficulties of preventing separation of these different layers as well.
So how handleable is that? If you want to say you want to detach the film and you want to roll this up, is that something that you could do in an upscaling approach? Is that feasible?
If you were to, well, first you would need to begin with patterning of a much larger structure, of course, not these little glass discs I have. But you can remove these effectively through a capillary force. So if you dip the glass slowly into an aqueous medium, it can peel it off if you do it precisely. So you could effectively peel off a sheet of this.
That's interesting. When we talk about biocompatibility, I was just, when you showed us the slide with all the different sources of the polysaccharides, you also had the shell, or
the crab?
Seafood, crab. It's known that if you, in chitosan, for instance, one of the reasons that people are going to mushroom is because some people are allergic to crab, to crustacea. So is that something, I've never thought of that. I haven't considered that. Would there be a possibility of something like that also happening for polysaccharides?
I think the issue there is specifically the purification of the material. And often when you purchase chitosan, it can be impure. There's a lot of remnant protein fragments or things like that. And this is what causes the issue typically.
All right. And is that maybe an application for this type of technology, the bacterial growth? Because you get now implants that have problems with bacterial growth, and so people are looking at different surface treatments. Is there evidence somewhere in the literature that kind of this topography could also stop bacterial activation or growth?
Yes, I believe that is possible, but I think it would depend on, do you mean just purely through topography or through?
Yeah.
Purely through topography.
Yeah. I believe like there is this biomimetic approach based on shark skin that covers the inside of your fridges to stop bacterial growth, if I remember correctly.
Okay.
So something like this might then also be possible with your...
I imagine then you could also produce something like that, yes.
Okay. Moving on then to the regulatory aspect. How do you see something like this work? For instance, from the technology point of view, well, the setup that you used is a simple one, so you had some variability, which of course is not ideal if you want to go to regulatory. So I presume that this is good for a thesis, but not if you want to upscale. So some of the technologies that you discussed also with Professor van Pyyvelde and that you mentioned, those would get rid of that variability?
In this case, there are rather expensive but available devices to perform nanoimprint lithography that are much more controllable. And they're typically used in a clean room setting. So you could effectively remove a lot of the variability that comes from me physically patterning all of these things on a hot plate and with that part of the procedure. I imagine there would be a bigger issue in a regulatory sense of the modified polymer itself. And so there would have to be quite an extensive study on that to see if it is safe to use in that setting. And
the implications on cost if you move this to a clean room setting? I imagine things become quite a bit more expensive then.
Initially, yes. Yeah, they would initially be quite a bit more expensive, but you don't necessarily need to have those devices in a clean room. They're just typically there because they're used for patterning electronics. So they need that clean room setting, but you could have them in a culture hood or something like that, for example.
Okay, so you can, if you just produce the materials, you can sterilize them sufficiently so that they can be produced in non-GMP settings then?
Yes.
Okay.
The materials aren't generally the issue of the sterilization. It's the setting in which you're carrying out that patterning.
Okay. And sterilization methods will not have an effect on either your material or on the patterning?
They could, depending on what kind of sterilization you're using. But I haven't had a chance to look at, for example, if you're using radiation or something like that to treat it.
Okay. And one thing that in tissue engineering is or one strategy that is coming up more and more is developmental engineering. So where you basically try to mimic development, and that means that you let the cells do what they want to do. So yeah. They talk to each other, they produce their extracellular matrix. Do you see a role of what you are doing in such a setting?
Potentially, if it was used in a way that could perhaps kickstart that sort of behavior. But then it would be something that would need to, I guess, be dissociated rather quickly if they're to do their own thing. So then I would have to look at the rate of degradation of these materials, for example, and how readily cells can actually interact with this material in that sense.
Yeah. Okay, and then I have one last question. Both your co-promoter and some other people here in the room are very much involved in silico modeling. Do you see a role of in silico modeling in your work? And what would that role be?
If you could accurately model, I don't know how readily that can be done at this point, but the influence of various topographies on cell behavior, but I don't think that's quite so simple.
Well, if you want to do that in a mechanistic way, but there is kind of these metaryomics studies where they just do a whole lot of these experiments and then they let machine learning pick something out.
If you were to do it in this sense and optimize the platform, of course, and feed it in that grid-like data from before, then potentially you could, in an optimal setting, use the same kind of algorithms to figure out perhaps what kind of patterns are going on and what is optimal in a given situation. But that would take quite a bit of experimental work and data to back that up.
And if we want to go for the mechanistic model, then what do you think, from what you have seen, would be one of the key issues to address to try and understand better the mechanism?
I think the key issue is the control of the properties in the material, and that is, I mean, I believe I did quite a good job at controlling them, but it's still not such a simple task. And you would need to carry out quite a bit of analysis to ensure that, in fact, you have the surface chemical composition that you intend to have, but then that can become quite expensive.
Okay. It's interesting. I was expecting an answer from the biological side, but it's a good one. Okay, those were my questions. Thank you.
Thank you.
Hi, Armen. I must say, initially, I was not thinking of asking any questions, but there was some pressure also on me to come up with a couple of questions. First of all, I would also like to congratulate you, same as Lies. I enjoyed very much your presentation.
Thank you.
I think indeed it was a very nice combination of being informative and entertaining. So a very good job. Now, I mean, if I look back at, let's say, the PhD track and I mean your project basically, and what we discussed in the beginning of what could be the various research questions and what could be the interest. Of course, things have evolved along the way, and in the end, you may have come up with something that we did not think of in the beginning, or what could be the main thing that would come out of your work. And so I'd like to, let's say, bring us back to the initial discussions, and obviously also my own research interest. I mean, I know it's kind of cheap, but nevertheless, if you see now what you have done and what you have achieved, I mean, well, you know that two of my two main interests at this stage are cell mechanics and blood vessel formation. Mm-hmm. So with what you now have established and achieved, I mean, yeah, what could be an interesting application of this technology? I mean, the technology that you have developed, or it can also be more general. I mean, the application of polysaccharides or certain aspects of things that you have explored. What could be a next step, let's say, in let's say doing something, I mean, useful or interesting, either in cell mechanics or blood vessel formation, or ideally, of course, the combination of the two. Well, let's say ideally, I mean, to meet my interests. And if I like it, I might just write a project application on this.
So for the first part, for the cell mechanics, I don't know how reasonable this is, but you do a lot of your studies with the traction force microscopy. So I don't know if it would be reasonable to coat films like this on the surface of, I believe you're using collagen?
Sorry?
You're using collagen gels?
Among others, yes.
Among others, yeah.
Yeah.
To coat something like this on top of that in a very thin layer and have a topographical patterning and see how that kind of interaction would happen. But you'd obviously have issues of modeling in differences in that topographical influence on how you're actually reading the movement of those fluorescent beads, the fluorescent particles.
So actually use it as a substrate for doing a traction force microscopy experiment.
Yeah, or on top of the substrate you typically use.
Yeah. So what would you like to learn from this?
Because then you could get a better idea of what kind of forces being applied, for example, in certain topographies or in certain areas of these patterns themselves. In terms of the blood vessel development, I think it would be quite interesting to see particularly obviously the grading structures, but also the pillars. They seem to allow multicellular arrangements in a certain way, or to use these properties to force certain multicellular arrangements together and see if you restrict, say, a given number of cells, can you influence how the vessel develops in a way?
Yeah, I think both could be reasonable. I guess with the first one, yeah, I guess there could be an opportunity of, let's say, investigating. I mean, to what extent actually there is an overlap between the effect of what is considered to be topography, mm-hmm. But then at the same time, indeed, you might be modulating mechanics. In fact, I mean, and the way cells apply force, and so what is perceived as an effect of topography might actually boil down to a cell mechanical effect.
And then one of the other things I didn't obviously have a chance to perform, but one of the proposed reasons behind why these interactions occur is the adsorption of proteins to the surface. So it would be interesting to examine if there is a localization in that sense of protein absorption that is driving this behavior as well.
Yeah. No, I mean, interesting. Coming maybe an additional question or comment on, let's say, the applicability within let's say, vascular biology or the formation of blood vessel networks. Obviously, I mean, vessels, I mean, tend to grow again in 3D. Yeah. And then it brings me back to one of the questions of Lies. I mean, while I agree that indeed it's a way, I mean, your technology is indeed, or can be seen as a way of looking at multicellular organization. Again, of course, yeah, how do we deal with this limitation of then studying multicellular organization in a more or less 2D fashion while, so yeah, what is your, I don't know whether. I mean, yeah, what is your feeling on this? But maybe you say, yeah, well, that's actually the same question as what Lies already asked. So that again, it's maybe a bit cheap or.
It's a very similar question, but
yeah, exactly. I guess
the difficulty there would be if it were the case that this organization of the cells is based on these properties in a two-dimensional sense, and they are being restricted, for example, because of that protein adsorption. To translate that into 3D is a rather challenging task.
Yeah.
Because I'd imagine you'd have specific, well, pseudo vessels that you could grow the cells in, and maybe restrict them along that.
Yeah, true. Yeah. I had a second question, but that is maybe even broader or also more philosophical. So when I was listening to your talk, and I mean at different let's say instances of your talk, you were actually referring to, you could say, to complexity. I mean, the fact that this is so complex. I mean, if you referred to this review paper where they came up with more than 400 components in the extracellular matrix. I don't know, were these all proteins or?
No.
Okay, well, luckily.
Yeah.
Yeah. But still, I mean, 400 components, that's quite something. I mean, at another instance, you were saying like, okay, I mean, polysaccharides are actually, I mean, it's very interesting, but also lead to a lot of complexity because again, one monomer can lead to different ring structures, and who knows? I mean, would it also then lead to different? Well, I mean, behavior, also cell behavior. Imagine that you have different rings, and then you bring cells in contact with those different ring structures. Would it all matter or not? And so, I mean, the more I was thinking about it, the more I became desperate, and thought like, oh my God, how do we deal with this? You could say, you know, I mean, from a fundamental basic science point of view, isn't that great? Because we can all explore those things, write beautiful papers, etc., etc. But in the end, yeah, what does it bring to us? I mean, because of course, it's very easy to get lost in this complexity. And okay, and despite all the beautiful paper that may come out of this, it may not bring us closer to an application. And of course, well, again, it depends on who you are and whether you say, I don't care about an application, but still we are, yeah, I guess most of us here in the audience, or many of us are engineers, or at least, let's say the jury members here that are present are all engineers. So I guess in the end, we care about an application. How do you, again, as I said, it's a philosophical question, so, but I don't know how you look at it.
Philosophically, in the sense of any application in tissue engineering, or?
Yeah, or how do we deal with this complexity? Because also, like for your method now, you explored, well, you could say two components, dextran and palmitic acid. You played with different topographies, basically 3 different topographies, but that by itself already leads to a lot of combinations.
Yeah.
And again, you could say, from a fundamental point of view, this is all interesting because I can see how, for instance, yeah, like the endothelial cell that you used, how they respond to it. Fine, but then what?
Yeah. That is one of the things that I was asking myself as I was doing it, and as I was reading research in this field. It seemed to me that we're taking very slow strides towards perhaps something.
Yeah.
And I think the difficulty, or overcoming that difficulty, is also not a simple question, because anywhere you look in this environment, there is a form of complexity on its own. So whether it's the composition or the structure or what have you, and I think I don't think we have an ideal approach towards this, and I don't know if there's an easy way to bypass that, or to look at it in a perhaps more holistic manner, as they do in a bioinformatics study, for example, but with properties in combination with different chemical compositions and so forth. So I don't have an answer to that.
Well, I don't have an answer either. Anyway, thank you.
Thank you.
Okay, thank you very much, Armen. I will leave some nice words for the after the deliberation. I was also asked to ask some questions, and I will do that. So the first thing comes to my mind is when you did your literature review, you came with this message that there's not much chemical modifications done, and people are using this on polysaccharides on shelf. They buy it and they just use it. I would like to know, what do you think is the reason? What are the reasons that there are so little rational design in the application of polysaccharides in this very challenging multidisciplinary field of tissue engineering and regenerative medicine?
Well, I think the simple answer to that is that the simplistic use can be very effective. And so people can effectively run with that. You can take a material off the shelf and show that it has an impact or a beneficial influence in some 3D conformation on cell development or cell growth or what have you. So it's very easy to run with a simplistic design of the material and see how far you can take it biologically. But I feel that approach isn't a very good one, because I'd imagine you'd run down to problems down the line, because you have very little control over what you're putting into this material.
Mm-hmm. And how do you think that we, for example, in chemical engineering would enhance this control? I mean, this topochemical engineering issue is one of my research passions, because it's how we can control the three-dimensional space and combine space and chemistry and interfacial interactions and so on. Mm-hmm. How do you see that engineering can contribute to improve this?
I think that would have to come through the development of methods for more precise chemical modifications at a greater scale than they can be done currently. Because I believe that kind of synthesis is quite expensive and time-consuming and tedious if you're to assemble units of a chemical one by one. You have to protect and then deprotect and keep going on and on until you have the structure you want. So it may perhaps be in the development of procedures to do that engineering could help.
Okay, and then I look at your presentation. It is my final question. At the pictures you have there, where you see that the cells, they are following this orientation in the structure there. I was wondering that the cells are intelligent. They have some sort of intelligence, and I was wondering what would happen if we take these cells out of this after some time and put them in another, let's say, flat surfaces. Would they follow the same behavior that they have there? In other words, could the experience of the cells in this small jail you have them for them would affect their behavior when you take them out?
I think it depends what cells you're putting there and how long you're leaving them there. For a short period of time, I don't know if a cell is going to, say, retain information with what it's interacting with. So for example, if you were to remove these cells from these surfaces and then culture them on the typical fibronectin-coated surface, I don't imagine they would suddenly start aligning with each other because of that. But if you had, for example, a stem cell that you forced down a specific lineage, then it could have a lasting influence.
Okay, thank you. I'm done with my questions.
Dames en heren, geachte collega's, ik geef u kennis van de uitslag van de beraadslaging van de examencommissie aangesteld voor de proef van dokter in de ingenieurswetenschappen. De heer Armen Tchobanian heeft aan de faculteit een proefschrift voorgelegd, handelend over Polysaccharide Esters for the Topochemical Design of Biointerfaces. Nederlandse vertaling Polysaccharide Esters voor het topochemische ontwerp van Biointerfaces. En dit tot het verkrijgen van de graad van dokter in de ingenieurswetenschappen. En hij heeft dit proefschrift in een openbare zitting voor de examencommissie verdedigd. De examencommissie heeft vastgesteld dat zowel aan de wettelijke en decretale bepalingen als aan de voorschriften van het universiteitsreglement ter zake werd voldaan. Na beraadslaging verklaart de examencommissie dat de heer Armen Tchobanian voldaan heeft aan alle eisen voor de proef en de graad van dokter in de ingenieurswetenschappen behaald. Bijgevolg, en in naam van de rector van de KU Leuven, wordt de heer Armen Tchobanian gepromoveerd tot de dokter in de ingenieurswetenschappen richting chemische technologie. In English, the most important sentences. Mr. Armen Tchobanian, the examination board has concluded that you have fulfilled all the requirements laid down in the university regulations for the doctoral examination. Therefore, on behalf of the rector of the KU Leuven, I confer upon you the degree of Doctor of Engineering Science, PhD in Chemical Engineering. Congratulations.
Thank you.
De zitting is gesloten. You can all sit down now, and Professor Pedro Fardim will say a few words to the new doctor.
Dear Armen, dear colleagues, it's a special day for us two today. So Armen is my first doctoral student in KU Leuven, after so many students in Finland. And our journey is also very special, Armen, because we both had a transition together. I mean, my transition from Finland to Belgium, and you were the first to come in and to face all the challenges that we had to go through the whole process of not having division, of not being in having a lab ready, and then finally to be able to bring a thesis like you did so successfully. And finally having to have the thesis in this condition, we are using masks and so on. So during the whole process, what was very impressive for me was our resilience into all the challenges that we have, the challenges we have in research, but also your openness. So you are not a chemist, and you managed to do the chemical derivatizations very nicely and the characterizations very nicely. You were always very open. You were not resistant to the suggestions and to what directions to go. If I look at your contributions, I mean, these two papers, this review paper is really interesting because of what you are bringing to the polysaccharide community. The message that you brought to the polysaccharide community is very relevant, and we see by the number of citations that is increasing quite a lot. And also the interest that this new research is bringing, because with the research you have shown that you can actually do the shaping of a polysaccharide, and cells can survive in these shapes, is quite interesting for other people working with 3D printing and other applications and so on. So I would like to warmly congratulate you for these efforts. At the same time, I would like to thank my co-promoter, partner in crime, with Hans. We just met, and in a walk when we have started the Leuven Chem and Tech, and I say, we have to work together. So it was just a click chemistry, as we say. And then we had an opportunity to share the work with you as a doctoral student. And we had a tough time in selecting the candidates. We had more than 50 applicants. And then Hans was the one who said, no, I like this guy. let's take this guy, right? So it was, you like him, man. So it was a very nice choice. So thank you for this nice collaboration. I would like to thank the members of the supervisory committee. It's a very nice experience for me also to see this possibility that we have in Leuven of having a supervisory board, supervisory committee with us that can follow the whole process, can give insights in the whole process, and then can be here in this day. So thank you so much, Liesbet, Geris, and Pedro from Pyyvelde. We also have an honor to have our decan in our supervisory board. We didn't know about that 4 years ago, but that is a good surprise in life. So I mean, I really appreciate the fact that you left Canada to come here to Belgium, to Leuven, and I hope you bring in your heart very good experiences from here, both for learning and growing, and also with pleasure and joy that you could find here. Thank you very much.
Thank you. So I think that's about wraps it up. I want to thank all of you. Unless I have to do something else. No? I can do it again if you want. But thank you all for your attention. Thank you all for being here. All of you who are joining us online. And you're free to go.
I have something to give you, but I first need a signature from you. I have a pen. You need to sign for the receipt of your diploma. Yep, down there. And then get from me. So, no more waiting. 2 years before you get it, so yeah, a long time. I don't even remember how long. No, they started to do that up front, but that's why I need this. You only get one. If you lose it, you don't get another. So, or if I lost it, then I would be in trouble.
Thank you.
So congratulations. And are you going back to Canada now? Or?
I have to leave at some point.
Your contract is out.
But I'm not sure that's the plan.
Right. Sure. Cool.